We have Dr. Mosier, of course, here hosting, and he brought on Dr. Meredith McKean, who I believe, Dr. Mosier, did you guys attend school together? Med school. Okay, so they've attended med school together. They're good friends. They talk a lot together. I've had them on the podcast, webinars. They are an excellent team, and we are glad to have them here. And then we also have Dr. Memi, who I just have had the fortune of meeting today. And we are so glad to have you here. I'm going to go ahead and welcome our speakers up on stage as I introduce you guys. So just by way of introduction, Dr. Mosier is a medical oncologist here in Scottsdale at Honor Health Research Institute. And he is, of course, our hosting doctor here from Honor Health. And he's currently actively developing new clinical trials and treatment options for patients with uveal melanoma. Dr. McKean, hailing from a one-way flight and a one-way out later from Nashville, she is our main Midwest board-certified medical oncologist, the director of the melanoma and skin cancer research for Sarah Cannon's Research Institute at Tennessee Oncology. And so we're so glad to have her here with us. And then Dr. Memi is a hematologist-oncologist from Santa Monica in California with over 16 years of experience in the medical field. So hematologist, which I didn't know this phrase until maybe 2 years ago, that's a blood doctor, like a blood doctor of some kind, right? No? Am I wrong? I'm probably wrong. What am I? Okay, hematology, he's going to explain. I have it wrong. So let's go ahead and welcome our speakers to the stage.
I think really the goal of this session, every session, is to be able to, offer information, but really use this as a platform for you to be able to ask any questions. So I think I'm kicking us off, but there may be a little bit of overlap, so my colleagues can feel free to chime in. So our session is really just how to make sense of it all. And I wanted to start and just kind of lay a framework of what drug development looks like. Because I think for patients, you're getting involved at kind of any place within that blue box, but I think just to be able to see the entire spectrum of the process, that before we even offer a patient drug on a clinical trial, it has generally undergone years of basic research, drug discovery, preclinical work in animal models, sometimes healthy volunteers, that has all led to an application to the FDA to prove why we're researching it, that this looks like we have some idea of the potential side effects and this looks very manageable, and then starting phase 1 through phase 3 clinical trials. I think as we'll discuss today, even what a phase 1 clinical trial means has changed a lot because the goal has really been, if there's a successful, a beneficial drug out there, we want to try to move it through the drug development process as quickly as we can so more patients benefit. And you can see that the median time from patent filing to product launch has dropped over the last decade with the goal of just trying to get these drugs to patients. And so this just outlines, phase 1 in general, the goals are looking at safety and understanding what dose we'd want to move forward with. Phase 2 historically has meant you've decided on your dose, you've decided on your patient population, and you're further exploring that. But as we'll discuss today, that's actually getting tucked into phase 1 studies now, with more so as a phase 1B. And a lot of that can just be speed and just being able to, hey, if we have a signal, especially for uveal melanoma patients, we don't need to close down a study to reopen necessarily as a phase 2, but try to explore this within the phase 1. And then a phase 3 is your more historical randomized clinical trial that patients are randomized either to receive therapy versus a standard of care. This is then reviewed by the FDA and then into phase 4 studies that even once a drug is approved, the FDA is still monitoring any safety events and looking to see are there any other long-term outcomes. I thought this slide was interesting. A lot of patients, when you're trying to decide whether or not to participate in a clinical trial, What this demonstrates is the change that I think we see as investigators and maybe patients see when they come in and they'll say, well, wait a minute, I haven't heard of this company before. I tried Googling this. And what this is showing, there's some acronyms here, is that more and more development is getting done by smaller biotech and biopharma companies. And then oftentimes either they're able to take that drug all the way through, or they're then partnering with a larger company that has maybe been around longer or has maybe more of the funding and resources. And so I always just try to tell patients that this is just kind of how drug development is changing, that we're just seeing more and more of these kind of smaller biotech companies. And I think in uveal, that's so important because sometimes these are the companies that are really willing to dive into rare diseases and really try to help us with patient outcomes. So just kind of going back through that process a little bit. So these preclinical studies, so the idea here with some of the animal models is determining the optimal formulation. Is this going to be a pill? Is this going to be IVs? Is this going to be a sub-Q injection? Try to decide the initial safe starting dose. For phase 1 clinical trials, oftentimes not oftentimes, we always start at doses lower than where we think the target is to make sure that we're starting at a safe level. And so that's decided based on the drug levels in those preclinical studies. And then looking at toxicity so we can understand, hey, based on this mechanism of action, these patients may have a risk of eye toxicity, so we need to make sure we're doing eye exams on the study. There may be a risk of cardiac toxicity, so we need to do echoes at baseline or on study. And so that's where the preclinical studies help us to be able to keep patients monitored as closely as possible while on study. And so then that kind of falls into then the clinical monitoring, whether that's EKGs, echoes, eye exams, and such. And I always have to remind patients that any clinical trial, phase 1 through phase 3, has had to go through FDA clearance. So this is submission of all the rationale based on mechanism of action, potential side effects, that is then given the okay by the FDA to move forward. And that's always important. So then once a phase 1 study initiates, the three of us could probably talk all day about just the different designs of a phase 1 dose escalation, but this is just an example of what it looks like to go kind of slowly up in dose, and any of these clinical trials can be further expanded once we've decided kind of what dose to proceed with. And one example of a phase 1 clinical trial, this was the initial clinical trial for pembrolizumab or Keytruda. And you can see here that basically within a phase 1 clinical trial, the first in human was initiated back in January 2011, got breakthrough therapy designation. There was a melanoma approval in September 2014 and lung cancer in 2015. This is a good example of how you can, if a drug looks really successful, historically this would have taken like a decade to get through, but just trying to get as much information in a phase 1 trial. And you can see how complex this is and how many patients were treated in a phase 1 trial. So a phase 1 trial doesn't always mean, and I think we're all very straightforward with patients. If you're the first patient treated, or if you're within the first 10 patients, it can be 1,000 patients treated on that clinical trial. And then my last slide is just helping patients understand when, for phase 1 clinical trials, we do a lot of extra blood draws, and this is for pharmacokinetics. And so the idea is, as you can kind of see in this image, we're really trying to understand what is the absorption, the metabolism, where is it distributing? And so this is why it is important for us to be collecting serial blood testing over the course of maybe the day. Patients have to come back in potentially the next day or 2 days later, because we're really trying to understand how long does that drug last in the patient's system, and how does that affect dosing and potential side effects. So I think those are just a few slides for food for thought, and I'll hand it over to my colleagues. Unless there's any questions.
Hi, good morning, everybody. Justin, thank you for having me here. It's a really great time to be part of this process. And Meredith set me up really nicely for about 10, 20 slides that I have. And I wanted to talk about, what's available out there now that Meredith's talked about what the clinical trial process is and what are different phases of clinical trials and how do we move from one phase to another. Does every trial move to next phase or not? So I think these are some of my disclosures. I wanted to talk about, some of the different targets in uveal melanoma in particular that we're trying to use to come to the next therapeutic option. So we're just going to talk about this sort of surface marker on the uveal melanoma cells called PRAME and how can we target that for benefit. Also, I think some of this talk already happened, trying to get to the metastatic uveal melanoma, these masses that are sitting either in the liver or somewhere else, can we inject those with different drugs to get a response? And there are a couple of different trials that look very exciting. And also target therapy, target therapy in the other cancer worlds, in non-small cell lung cancer, breast cancer, melanoma, it's quite well known. I mean, the first target therapy was for CML with imatinib or Gleevec, and that's been the dream in every other cancer. Is there a silver bullet for a mutation that's present? And so, again, we're looking at that for uveal as well. And then also in the last about 5 years, even though the cell therapy is as old as, before I was born, but in melanoma, in uveal melanoma, it's a quite new concept, and it's especially in cutaneous melanoma, it's an FDA-approved therapy. So, is this a viable option in metastatic uveal melanoma as well? So I don't know if I can. This is a hyperlink. I don't know if I can get to that or not, but this hyperlink essentially was for clinicaltrials.gov, and if you select certain parameters, you can come up with a list of clinical trials, and it could be quite cumbersome to know which trials are recruiting, are closed, ongoing. So when I put in for metastatic uveal melanoma actively accruing, there are only about 23, 24 trials. So again, disease space is small, and but in metastatic setting, there's still not that many trials that are going on. Okay, so we're going to start off with the phase 1 study from Immunocore of this drug called IMC F106C. I think my colleagues know this really well. It was presented by my senior partner, Dr. Hamid, in Europe a couple of years back, where we looked at all different solid malignancies that expressed PRAME, and they were of particular HLA subtype, the white blood cell sort of marker that Dr. Moser talked about a little bit earlier. And some of these patients that were treated, they had uveal melanoma as well. Either they had previously treated or not. And we see some signal there. So this is one of the possible options moving forward. And it's up to us as advocates for our patients and the patient advocacy groups to push these companies to consider these trials further. Because I don't know where the Immunocore folks are going with the PRAME targeting in uveal melanoma. But this is one of the options, and I've treated more than a few patients on this study. I think it potentially is a reasonable option moving forward. I'm not going to go to too much about the response rates or the RF's benefits. I think we could talk about that on a separate day, but just wanted to show you that there are some options targeting different of these. And then Immunocore drug is a bispecific antibody, a different option to target the PRAME, and it attracts the T cells, the effective T cells that we want to generate antitumor activity. Another way is to generate a lymphocyte that can target PRAME. Similar mechanism, but using a different way to do it. Again, it appears that this is a viable option for metastatic uveal melanoma. This is one of the Plume trials that's looking at that. Again, these are HLA-O2/O1 negative and positive patients that will be screened, and some of these are treated with Kimmtrak and some are not, and they will be then looked at with pembrolizumab with lenvatinib, so one of the other studies that's going on from a targeted standpoint. This is one of the drugs that was a part of when I came out of my fellowship, and we looked at cutaneous melanoma that wasn't responding to immunotherapy, or head and neck cancers that weren't responding to immunotherapy. And this molecule, SD101, is sort of a short a piece of DNA that is not methylated, and our body recognizes unmethylated DNA as foreign, as a bacterial component. And because of that, it mounts an immune response to that. So when we injected these short pieces of DNA into the tumor, it created an immune response to that tumor. So this data is already published in head and neck and cutaneous melanoma, and then the company was small. And one of the things Meredith was talking about, a lot of these companies that are doing this early research are small, and sometimes, those companies can't be viable. So this company originally went down, and the molecule was acquired by a different biopharma now, and they're taking it through uveal melanoma space. And what they're doing is they've sort of, when I was doing this cutaneous melanoma work, we injected the tumor. Now what we're doing in this particular trial is exposing the liver under pressure with this drug. And that's been really helpful in providing higher doses of this drug to that area and see if we can improve the outcomes. And this is one of the reasons we think it works, is because it suppresses so it's kind of like a double negative, suppresses the suppressive activity of immune system on itself. So it's sort of, there are accelerators for our immune system, and then there are brakes for our immune systems. So this is kind of breaking the brake. So these myeloderived suppressor cells are the brakes for our immune system. They don't allow anti-tumor activity that we want. So trying to regulate them, trying to decrease their numbers, trying to decrease their activity, hopefully will improve the anti-tumor activity. So one of the options that's available, this is the trial design. And again, another way to sort of activate the immune system is trying to inject it with a PV10, which is a molecule called rose-Bengals disodium, and it's an oncolytic intralesional therapy for the liver metastases that again activates the immune system in turn. And you can see in the waterfall plot that there's a nice responses in very good number of patients. And then I talked about the targeted therapy. Gleevec came out, and everybody was dreaming that there's got to be a Gleevec similar for all cancers. And in some we did, in some we didn't. And more recently, we know that uveal melanoma has some mutations that perhaps are activating 11-GNAQ, is Dr. Moser talked about it a little bit earlier in the NGS talk. It subsequently activates the protein kinase C pathway, and we know that protein kinase C pathway is more than reasonable target for disease control or cancer control. But by itself, it may not be sufficient, so it needs another partner. Just like in skin melanoma, you have BRAF mutations that takes 2 drugs for better control than 1. Similarly here, the darovasertib drug is being combined with crizotinib to get a better control over disease, and the data looks very promising. Early data has now led to phase 3 studies that are open at multiple sites, including ours. Some of my colleagues here have been at the forefront for development of this drug. Again, safety and efficacy, whenever you're combining, like Dr. Moser was saying earlier, we like more drugs to be sort of, to give us a better control of cancer, but that also leads to some toxicity. So there are a number of, safety concerns with darovasertib and crizotinib combination, but most of those issues are fairly easy to manage. And the last thing that I can talk about is tumor infiltrating lymphocyte. And a little bit about that is, what is TIL? So all tumors have these immune cells, or most, maybe not so much true in uveal melanoma, but most of the tumor it's not just cancer alone. It has other components, other cellular components that could be immune cells, fibroblasts, other supportive cells. They are either helping or hurting the cancer one way or another. So in skin melanomas, we know that there's a huge component within the tumor itself of immune cells. And for one reason or another, they're stuck. They're not acting towards the cancer. So one of the ways we can sort of improve that process is, can we increase the number of these tumor-infiltrating lymphocytes and give it back to the patients and see if we can control the disease. And in cutaneous melanoma, patients who failed frontline Keytruda, Optivos, or the combination drugs, they are candidate for this therapy, and it's FDA approved now. Now, can we replicate that in uveal melanoma? We know that the uveal melanoma doesn't have the same kind of mutational status as cutaneous melanoma, but there have been a number of trials that have looked at, is TIL therapy a viable option for metastatic uveal melanoma? And we're seeing early signals that yes, it's possible. And one of my favorite uveal melanoma oncologists, Dr. Patel, has done a lot of work in this space, led the work in this space, looking at the TIL therapy that's already available, or working while she was at MD Anderson, working with the researchers to see if we can improve the TILs that we're getting from these tumors and improve the efficacy, improve the numbers of TILs we get, and that could potentially sort of result in a better response. So TIL 1.0 versus TIL 3.0, we're able to get more TILs from the primary tumor, grow them, and perhaps, give it back to the patient and have better results. Okay. Again, showing the same thing, that it's a more than reasonable option to control the disease in advanced setting. Okay. I'll leave it here. So a lot. I didn't want to get into too much nitty-gritty of the data. Just wanted to, after Meredith's talk, that there are options, and the best way to consider those options is talking to your oncologist and see what makes the most sense.
Sorry I joined the session late, but could you possibly go back about 3 slides to the different TIL therapies? One of those TIL therapies is marketed and it has FDA approval called
Right, so that's the Iovance's drug.
Which one of the trials? Can you go back one more slide maybe? Yes. I can't pronounce the name of the drug.
Lifilicel.
Yes. Which one of these? Is it one of those 2 trials?
No, this is not. I'm not focusing particularly on Lifilicel. There is an ongoing trial at Moffitt with Lifilicel in uveal melanoma, but this is just looking at a number of different TIL therapies that are potentially available. It's a more centralized process through pharmaceutical industry.
So in question to lifileucel, so lifileucel was actually developed from the NIH, the National Surgical Branch, as you know, and they actually started testing lifileucel in the 80s. So when Dr. Mamie made the comment, this has been going on before we were born, he's right. It's taken almost 40 years to get the TIL therapy. So back at the National Surgery Branch of the NCI, they did do a study a couple of years ago of, I think, what did become lifileucel, but this was years before it actually became approved, of looking at this TIL therapy in patients with uveal melanoma. And I want to say that data showed a 20 to 30% response rate. So there is some data out there. It's just because with TIL, you have the kind of non-genetically modified TIL, which is the lifileucel, and we have a lot of the newer ones where they're tumor-informed TIL or genetically modified, and those are the studies that Dr. Mamie was highlighting, and those I think we have less data at this point in uveal melanoma. So I think those were some great high-level overviews about what is a clinical trial, what clinical trials are out there. Does anyone have any questions before we start asking the questions of how do you navigate this space, right? We just threw all this information out there. How do you navigate it as a patient? Okay, so I'll throw this to Dr. McKean and Dr. Mamie, but so as a patient who's newly diagnosed with uveal melanoma, obviously we have, within the clinical trials, there's first or second line clinical trials, and then there's clinical trials for patients who are refractory, so any line. So we'll start kind of earliest in the course. So with frontline trials, we obviously have the randomized study from IDEA, looking at the combination of darovasertib and crizotinib, which is led by Dr. McKean. And soon we'll have the Replimune study, which I believe is second or first line for patients. So when we're seeing new patients, how do you navigate, should we go on a clinical trial versus a standard of care? And also, how do you navigate the question that a lot of the first and second line trials don't allow prior liver-directed therapy? So when a patient asks you, well, I just have one spot, should I get it radiated? Knowing that I could not go on this trial in the future, or should I go on this trial? How do you answer those questions, and how do you start navigating those situations?
Yeah, I think first of all, it's great to have this dilemma now in uveal melanoma. I think we've been able to see the field evolve so much that to have options and to be able to kind of think through what's the best first step and second step shows how much progress we've been making. But there still is more limited treatments than we'd like, and that's why we do have to be really thoughtful. So I love this question. I think for patients, to me, some of it in the frontline setting is what's the disease trajectory? Where is your disease located? In general, most patients develop metastatic disease in the liver. And so the question is liver-directed therapy now versus later. Certainly a patient that the disease trajectory is pretty rapid, that might be a patient that you say, hey, should we be looking at liver-directed therapies first? HLA testing is always important to do upfront, both for trial inclusion, also just to understand is the one FDA-approved systemic therapy, Kimmtrak, is that an option for you or not? Now, I would put Kimmtrak and ipilimumab and nivolumab or Yervoy Opdivo in kind of a separate category because the advantage is you can do those at any time. And so, there's no restrictions versus several of the clinical trials do limit, hey, you have to have this in the frontline setting or the second line setting. And so then a lot of it becomes, if it's not a clear what's the trajectory of your disease and do we need to do liver-directed therapy up front, then it is kind of discussing, well, here's your systemic therapy options. And I do see clinical trials and patient participation as, the best outcomes we see is, I like to use the term of best shots on goal. We don't know. I wish I knew exactly which patient was going to respond to each treatment, but we don't know. And so the nice thing with clinical trials is being able to say, hey, well, here's another option to see if this is the best option for you, if this is going to slow down your cancer. And so especially for the studies that are frontline, this may be your only chance to get this drug. And so just discussing, hey, here's what the data that we do have, here's potential side effects. Is this something that you're going to want to try because we may not have access to it later? So it really is for the patient in front of you trying to discuss, hey, for your individual disease. Now, I think that Cure Insight does a really good job. I know Kevin's always summarizing all the trials available because it is really hard and can be center specific. And I do think as investigators, we do try to kind of collaborate and say, hey, what do you have open? I've got this patient that, this is kind of what I think might be the best option. It might not be available here, and try to direct patients, if they're able to where they might go for their best care.
I absolutely agree with what Meredith has said. I think being in the space or the area where I practice, I see refractory cases or patients who haven't responded to their previous treatments. That sort of changes my perspective a little bit when I answer this question. I favor clinical trials over any standard of care therapy if the patient's able and willing, right? Because like Meredith said, you've got one shot at goal. Like some of these patients that I've seen with metastatic uveal melanoma, if they had gone with liver-directed therapy, they would not have had the shot of being treated with drugs like bernetofusp, which is the PRAME targeting drug. And they haven't had the long-term benefit they're having, right? So, and then even these are the same patients who are candidate for Kimmtrak as well, right? But Kimmtrak doesn't give me 100% response rates. Kimmtrak doesn't give me, long-term durable control of these disease. So what can I, based on the data that's been presented at different conferences for these drugs, which gives me the most confidence? So that's sort of the approach that I take. But at the same time, I think if we don't have the courageous patients that are willing to take part in these studies, we won't be here having this dilemma of, hey, these patients aren't able to get on these trials and this and that. I think, and also talking about, liver-directed therapy itself, for a very small number of patients, that's the path to long-term control. that means majority of patients will need systemic therapy, right? Or treatment of the whole body. And right now it's tough to tell which patient is that, the one who's going to have long-term control with just liver-directed therapy, or will they need something else? So my usually goal is, if a patient's coming in, I got to put them on systemic therapy because they're going to need it. Now, patient's life, patient's choices, they matter immensely. So that discussion needs to take place. But my idea is, if I can get a patient on clinical trial that at least promises me benefit as much as what we have as standard of care, that's usually my goal. And that obviously that's a frank discussion between my patient and myself. So
I think that's very similar to my approach as well. I use the analogy of tools, and I think in general we don't have as many tools to treat uveal melanoma as we do other cancers. And so my approach typically is, what is the best treatment path that's going to give you access to the most tools? So when it comes to the question of, should I do a liver-directed therapy versus a frontline trial that would exclude me in the future if I got this, I typically recommend the trial because that gives you the option of that tool and keeps the toolbox full. Now, you have to balance that with everything going on. I had a patient the other day where we were in the discussion of, okay, we know Hepzato is a great approved option for patients with liver mets, but you have to have less than 50% disease in your liver. And we had someone who was in the 40th percentile in terms of the disease in the liver. And it was, do we try this trial, knowing that we may miss the boat for Hepzato if it doesn't work, or do we try Hepzato knowing you're not going to be able to go on this trial? And sometimes you're in those situations, and then it's just a really frank discussion with the patient. But in general, I think it's all about keeping the toolbox full and keeping access to the most number of available therapies, since they are so limited for patients, unfortunately.
So I have a question. I am a patient as well. I'm a metastatic patient, just for those of you who don't know. And one of the things that I talk to just a lot of the biotech companies about is how it can be really frustrating sometimes to be in a situation where maybe you don't need full body treatment. You only need to treat one or two. And that gives you a good duration of time. Is there room in the discussion, I guess, between oncology and clinical trials, primary investigators, to push for patients to be able to have access to a trial, even if, say, tumor one that was singularly treated 3 years ago, let's pretend, that'd be ideal, but that doesn't happen. Let's just say it's been a duration of time, they were treated, now they have new lesions, and they want to go on that trial. I'm hoping this is making sense. Is there room for that discussion of, can we push for their entry to the trial with the new disease, and how can we, I don't know, and maybe pharma reps, maybe you guys can answer this a little better.
Yeah, that's a complicated question.
I know, it's so complicated.
And I think we're starting to see some of those. I believe the randomized study that's coming out within October later this year and early next year does not exclude patients with liver-directed therapies. I hope I'm not saying that wrong. But a lot of it comes down to the requirements needed to get the FDA approval, which Meredith is probably involved in more of these discussions than I am, but that's really the challenge, right? We can do a trial that encompasses everyone, but we also need to have a very clean patient population to study for the FDA to look and say, is this truly better? Because if you have a very kind of diverse patient population and you're trying to compare it to a very clean patient population, we can't always make
the data gets messy.
Yes.
Yeah, okay, that makes sense.
That's where we're kind of trapped. I don't know if you guys have any other comments on that.
Yeah, I agree. I think as investigators, we're like the number one patient advocates, and we're always pushing to try to, push the limits on what is allowed. And I think the FDA has put out several white papers, trying to say not only from an inclusion exclusion standpoint, but even just where can you get safety labs, right? Clinical trial participation is still really limited by patients having to be able to travel to major centers. So I think that there's a lot of push up front. The challenge is once that protocol is set, we have to abide by it. Yeah. There's no wiggle room. I wish there was, because I think those are the hardest conversations that we have, is when we're so excited about a trial for a patient and there's just one little glitch to whatever the inclusion exclusion is. But that's something that like we've agreed with the FDA, and like there's just no wiggle room there.
So it almost, I mean, it's again still a complicated question, but it almost sounds like maybe if back in the inception of the trial idea, that before it even goes to the okay, now you're ready to go for phase 1, we're ready to do the the investigation portion of the trial, that those trial criteria that they're submitting to the FDA, if they have the wiggle room, that's where they have to say we don't want to exclude if possible. Like that it would have to start there, like way back before the trial becomes a trial.
Yeah, I'll kind of start that and then pass it on.
And I could be totally wrong. That's
usually phase 1 trials are more encompassing, right? We kind of talked about, and I know I was in the FDA session, that actually phase 1 trials, that's sometimes an opportunity for patients with uveal melanoma to get enrolled because they're still looking at all solid tumors. Oftentimes it's as we move through the process and as it gets closer to FDA filing, they want, as Justin said, kind of the same patient population. And so that's where we try to push and say, well, look, it's been safe for, these patients. Can we continue to include them? But I don't know, as a phase 1 trialist, if you have other thoughts on that.
I'm of the thought that the trial protocol process is very archaic. It's the same stuff we've been doing 20, 30 years ago that we're trying to replicate with the newer drugs. I'm not going to be able to change that. And I think all of my generation of physicians, as hard as they try, cannot change that. But in my opinion, there are 2 components to the trial. One is the scientific research part, and other the patient treatment part. Sometimes they don't kind of go together. For the scientific research part, you want as pure of a population that you can have to get the signal to move forward.
Yeah.
From a treatment perspective, for a patient, I want that tool.
Yeah.
Right? And sometimes that patient may not fit that population that we want. And that's where the discohesion comes. And that's where we, get frustrated that I want this patient. And there's no sort of, there's no practical parallel process to get that drug to that patient. I know there's compassionate use stuff, there's right to try. It's not very practical.
Yeah.
I don't know if you can hear my frustration.
No, I hear you.
And I think with that too, I think the companies are on our side. I think they want to be as inclusive as possible. But you're right, Indrajit. Like we're following these approval processes that have been very similar for many years, and obviously the drugs and how we work and how we can monitor responses have really changed. And I think we're starting to see some of that change. There was a big group of melanoma medical oncologists led by Jason Luke, who just sent a letter to the FDA saying, hey, we think you guys got to update some things. And they responded favorably in a response letter. So I think we're starting to do that. So hopefully in the future we'll see it. But it is a lot of change that needs to happen to get that flexibility. And it's probably going to be years, unfortunately, but it is starting.
And this is what we talked about. If any of you guys who listened to the FDA session, I know I wasn't over there, but this is why we talked about that, right? That there's such a long process. Talking to the FDA to get anything changed takes time. Getting a drug to the FDA to get approved takes time. Taking the drug to market takes time. And that is not a fun thing to hear, especially if you're a metastatic patient. But the beauty of clinical trials, I think that we can kind of round this out with, is the clinical trials and the people who participate in clinical trials make it possible for that time to pass in, I'm trying to think of the phrase, like to use it well. Like that we're actually using the time well, and we're getting the research, we're getting the data to then move a drug toward approval. That's how we got to Dementifusp. That's how we got to Hepzato Kit. Like these processes do take time, but the results of all of the people who did those trials are why we have them today. Anybody else have questions from the audience? Dr. Hadfield, let's get over to you. You get to speak. You get the mic, because,
I could have done it without the mic. One question that I have about this, this is like such an exciting time to work in this space. There's obviously all these new therapeutic options that didn't exist 5, 10, 15 years ago. But the more and more I see the data and the more and more I'm involved and the more I see what's happening at my institution, do you think that we're going to have an issue in the near future where these therapies are going to get more and more and more siloed into larger academic centers, just given how complex they are to give, like TIL therapy, bispecifics that require CRS monitoring, other therapies that just will only be able to be given at large academic centers. Do you think that's going to be a problem in like the next 10, 15 years?
Sure. Yeah, you just like tossed it right to me because Sarah Cannon Research Institute, our whole goal is trying to get research out into the community. And I think we can all say the centers where we're at. So I actually think we're making a lot of progress here. And I think some of this is because some of the drugs that have been approved before, right? Now that TIL is approved for cutaneous melanoma, I think all the centers are now trying to figure out how to operationalize that, who's the team at these locations. I think with Delcath approved, you're going to see more and more institutions getting training and comfort to offer Delcath. So my hope is that, I think Uveal is still, this is a unique patient population. I think there still will be a need for kind of a specialized, personalized plan for each patient. But I actually think we're making a lot of progress in trying to get some of these treatments closer to home for patients, because that's how we're going to make the most progress, right? If we have the best drug available, but only 5% of the patients can get it, we're not really doing a service. So I think it's a great question, and I actually think we're making a lot of progress. I think like with KIMTRAC, the KIMTRAC Connect, right? They will help patients now that it's FDA approved. Hey, you can get your first 3 doses at kind of high volume centers. They'll help with transportation, get those kind of highest risk doses at these high volume centers. And then, hey, let's go out and train your local oncologist so that team feels comfortable doing those further weekly treatments. So I do applaud some of the pharma partners that have really been helping with this problem to really find a patient-centered solution.
I think I'm going to use the example of Opdivo and Yervoy. Like when it was approved, because of toxicity concerns, a lot of the community oncologists weren't favoring, and they were still using either Keytruda or Opdivo alone to treat their patients with metastatic melanoma, from skin melanoma at least. And as the word got out, as expert medical oncologists in melanoma world went and talked to these folks in the community, the confidence to treat with Opdivo and Yervoy built. Right now, I see patients that are refractory to Opdivo, and those are difficult patients to treat, but I'm glad that these patients didn't have to travel, from Pasadena to Santa Monica, a 3-hour drive, just to get their 1 dose every 3 weeks. It's a 30-mile drive, but it takes 3 hours, right? Let alone going from one part of Arizona to Scottsdale and whatnot. So I think with time that will come. Right now, I agree. Like I see a lot of the patients, the first-line patients for metastatic uveal melanoma, is because they're HLA, positive and they need KIMTRAC, but the local oncologist isn't comfortable, or they don't have the logistics to set up, in a small hospital to monitor these patients for CRS. They have 3-bed ICU in middle of Central California. So that's not viable. So they'll come to us, we'll do the first 3 doses. It's still painful. These patients are driving, they have to stay in expensive hotels in LA. But I think as the time goes, just like Opdivo and Yervoy, things will get better. And I hope it's not centralized. I really hope it's not, because then you're excluding a number of patients from these therapies.
I'm Kevin O'Neill. I'm in Philadelphia. I'm a 5-year survivor and still moving on. So I just wanted to take your point and take it one extra step. There are probably a dozen centers of excellence for medical oncology in uveal melanoma, including your centers that you're from here in Arizona, in Los Angeles, in Nashville, on the East Coast. But that's just a dozen centers, and that means if someone wants access to these clinical trials, they're likely going to have to travel. Meredith McCain, you're in Sarah Cannon in Nashville, and you're also affiliated with centers in Florida. And so probably 2 years ago, I had asked you for a contact at Sarah Cannon for colon cancer for a friend living in Florida, and found out that trial that you had at Sarah Cannon was in one of your colleagues' centers also in Florida. Can you talk about kind of this regionalization of medical practice where maybe in the rare cancer setting, some of these trials can also skip across state borders, so to speak?
Yeah, and I think that goes back to a lot of the conversations with pharma partners, right? And so a lot of the trials, so like the IDEA trial now that's phase 3, I think they've done a really good job of trying to get that out into a number of clinics, across the nation now to try to make it as accessible as possible for patients. So, one of the challenges is the early phase 1 studies. Those still do require, because we're trying to understand the safety, the levels, those will need to continue to be done at more specialized centers. But like one of the things that we do at Sarah Cannon is, okay, well, once we've found the dose, and even if it's phase 1B and it's still a phase 1 trial, but we kind of understand, we have a good idea of what the safety is going to be. We have a good idea of the dose. We're just enrolling more patients. Can we push that out to like the next level of clinics in the community that are familiar with these types of studies to try to offer it to more patients, maybe even, earlier, so these patients have access before they've had multiple lines of therapy and are having a lot of symptoms from their disease. And so I think that's really where we're trying to go in oncology, is trying to push them out as close as possible for patients. I think we're all part of kind of networks where we do try to not only communicate with other specialized centers of what trials are available, but then also how can we get this treatment as close to home for you as possible? Because I think everyone can agree sleeping in your own bed at night is probably the best medicine. So, having to travel, every week for, 3 hours one way is challenging. And so I think like for Sarah Cannon, we certainly do try to look at the network. Now that we have trials across most of the US now to say, ooh, can we get this closer for this patient? Or some of the models are, okay, well, maybe it's not open yet, but we actually have communication that if we find that patient, then we can open up at that site within 2 weeks. And I think that's maybe a model we'd probably love to do for every uveal study, but something that we're all working towards.
So I love that you guys are talking about like clinical trials, and just as we're finishing up before we go to our break, I guess I'll get back up here where you guys can see me. It's kind of like hearing you guys talk. It sounds like there's this kind of gradual graduation from level 1 to level 2. And obviously we have the phases and that's how it works. But I love that we've been able to break down some of the aspects, right? There's the travel things, the things that you guys as oncologists think about when you're sending a patient to a trial. So thank you guys for these thoughts and for everything that you have given and that you continue to give for your patients to make sure that they have the best care possible and the best chance at, like you said, like just having more time and continuing to see research evolve. So let's give them a round of applause. Thank you, guys. Okay, so next up, we're going to thank Immunocore. So Immunocore is a commercial stage biotech company that discovered, developed, and commercialized the world's first approved T-cell receptor, or TCR, therapy, KIMMTRAK. And our mission, or their mission, is to radically improve outcomes for patients with cancer, infectious diseases, and autoimmune diseases by pioneering and delivering transformative immunomodulating medicines. So we are so excited that they are here, and I can't tell, is Katie Wall, are you here? Katie, oh, Katie, hi. I talk to you a lot on email and the phone and all the things, and I think you also have Blake with you, correct? Yes, so Katie, Blake, raise your hands, patients in the room. They're over here. You guys can chat with them. They're your go-to to talk about KIMMTRAK Connect. Make sure that you know that you've had the right HLA typing testing done. Probably sound like I don't know what I'm talking about. My mouth is just a little tired. So you guys know what I mean. The HLA type test, there's all kinds of little nuances. We've had, I think, a couple of years under our belt as a patient population of trying to make sure that patients know what they need to talk to their oncologists about. And I think that the oncologists all around the regional centers that treat uveal melanoma are a lot more consistent now with making sure the HLA testing is done. But if you haven't had your HLA typing done, talk to your oncologist. It can be done. It doesn't have to be done because you're metastatic. And if anything, it should be done first, way before you ever have the potential or the possibility of metastatic disease. For those of you guys online, head to your websites. Those of you guys here in person, don't do this yet. We want you to stay here. But you can head to our registry and you can help further research, and you can also help find trials. One of the cool things about our registry is that we work with pharma, we work with our biotech companies to create surveys sometimes and to give them an opportunity to look for the right kinds of patients to participate in a trial. So if you want to be on those lists and you want to get an email that says, are you interested in a clinical trial, then you need to be part of the registry. You need to be registered. You need to sign your consents, and you need to start filling out the surveys that have information about your demographics, like where you live, how old you are. Those are important kinds of things, because if they're looking for a trial and they want to do a trial of people who are over the ages of 20 to 50, they're going to look in the registry and they're going to pull that data. So if you are not answering your surveys and telling us who you are, how your medical history is going, we can't give you the clinical trial